Hello, everybody. Welcome back. This lecture is going to cover Chapter 5, which is cell membrane structure and function. So in the previous chapter, Chapter 4, we took a look at some general characteristics of cells and all their different component parts. And one of those component parts was the plasma membrane, or also known as a cell membrane. So if you remember, some of the minimal requirements for something to be considered a cell is cell membrane, DNA, ribosomes, and some cytoplasm and cytoskeleton. So we're going to focus more specifically on the plasma membrane, kind of what it is made up of, and what its job is, and also how things move inside, into and out of the cell. Those are kind of our topics. All right, so what is it? What does it do? So again, plasma membrane, cell membrane, those terms are kind of interchangeable. So you could use either term, whichever one works best for your brain. So basically what it is, it's the barrier, right? It's that physical boundary between the components on the inside of the cell, all the organelles and cytoplasm and nucleus and DNA and all that kind of stuff that separates it from the surroundings. Now, depending on your type of organism, those surroundings could be quite different. So if you're an amoeba or a paramecium, a single-celled protist, the surroundings are the pond you live in, or the ocean, or the river, or the lake, or whatever watery environment you're in. If you're a multicellular organism, that environment will be what we call interstitial fluid. It's the fluid that surrounds all cells. So our bodies are about 50 to 60% water, and each one of our cells is bathed in this fluid. So no matter whether you're a single-celled organism or a multicellular organism, you have to have a physical barrier between what's going on inside of your cell and what's going on outside of your cell. Okay, so here's some of the functions of the plasma membrane. So just like I said, it isolates the cell's contents from the environment, whether it's a physical environment outside, like in a pond, or a more internal environment, like what would be inside of our bodies. It helps to regulate what moves in and out of the cell. So this is called exchange or transport or permeability. Sometimes you might see that word being used. So it is the regulator. It's like the gatekeeper. It decides what goes in and what goes out. Not like it has a conscious to do that, but its chemical properties are what decides what goes in and out. It can communicate with other cells. So you might have two cells come close together, and when their membranes touch, that can send signals and communicate to each of the cells. Whether you are an individual unicellular organism floating around, like a couple bacteria bumping into each other, or even within a multicellular organism, your cells are connected and they allow you to communicate via the proteins usually embedded in plasma membranes. There can be anchoring proteins. We're going to be talking about plasma proteins here in a little bit, but they can attach to other plasma membranes, especially in multicellular organisms, especially like in our skin. You have your epithelial cells tightly connected to each other, and their membranes are tightly connected to each other. And it also helps to regulate biochemical reactions. So we'll be learning about enzymes later. Enzymes are special types of proteins that do chemical reactions. And so sometimes those enzymes are embedded in the plasma membrane. All right, so what is it? It's a physical barrier that separates the inside from the outside of the cell. And what does it do? A lot of stuff, okay? That list of 5 we just listed there. Okay, so what does it look like? So here's a close-up picture. You have this in your textbook. It's the first one, figure 5.1. And so the accepted explanation for the structure of the plasma membrane is what we call fluid mosaic. It is fluidy and flexible, but it is not monotonous. It is not just uniform and homogeneous. It has lots of different things going on, and so that's the mosaic part of that term. So fluid it's flexible, it's modifiable and adaptable for different environments, and it's got a whole bunch of other component pieces in it, specifically lots of different kinds of proteins. So those are the blue blobs there. So the layer itself is what we call phospholipids. So we saw those as a type of lipid back in chapter 3. So these are the little yellow molecules right here. So they have them labeled phospholipid bilayer. So each one of these is a phospholipid. So if you remember, phospholipids are unique in that they have one side that's polar, which has a charge to it, and one side that's non-polar, which doesn't have a charge to it. And so that brings in those hydrophobic and hydrophilic interactions, like we were introduced back in chapter 3. Okay, so here's a closer look and a reminder of those phospholipid structures, right? So we have two fatty acid tails sticking off of the side with a charged phosphate head with that big phosphate functional group in there. So reinforce that idea, hydrophobic, water-fearing. They don't have any interaction with water because there's no charges or partial charges with the hydrocarbon tails. But because the phosphate head, that hydrophilic head does have some charges to it, some polar covalent bonds are in those molecules. It can be around water. It's hydrophilic. It has that interaction with those charged particles. So when these molecules orient themselves, they just do it automatically. There's nothing that's going to tell them to do this. If you're to get a bucket of phospholipids and just dump them in a beaker of water, they would automatically do this. That's just their chemical properties. So the hydrophobic tails would be pointing towards each other, and the hydrophilic heads would be trying to get around the water, and the hydrophobic tails would be trying to get away from the water. So this is what we call a bilayer. So when we use the phrase phospholipid bilayer, it's two layers of these phospholipids with their hydrophobic tails together and then the hydrophilic heads pointing towards the internal watery environment of the cytosol and the external watery environment of the pond or the river or just that interstitial fluid that surrounds all of our cells. Okay, so some more characteristics of this is it's flexible. It allows cells to change shape. I have a couple of videos that you can take a look at on that main page for this week's activities that you'll see an amoeba kind of engulfing some paramecium. So that flexibility of that amoeba being able to do that is thanks to this fluid nature of this lipid bilayer. They're not bonded to get together. They're just in this associations of the hydrophilic sides wanting to be close to each other and the hydrophobic parts wanting to be close to each other. So it's just these associations, if you will, or attractions. There's no bonding actually taking place. Now, if you remember when we were taking a look at lipids back in chapter 3, we had some different kinds of lipids, fats versus oils, right? So one of the characteristics of a fat is those fatty acid tails were all saturated with hydrogen. So there were no double bonds. There's this big, long hydrocarbon chain, and they were very linear, right? So you had your three fatty acids, and they're all right next to each other, so they could stack very tightly together, and that gave you a fat, solid at room temperature, right? As opposed to ones that are kind of kinked with, I can't bend my fingers in different ways, but if they had the double bonds, if they were unsaturated fats, then they had some kinks in them and the lipids could not get very close to each other. Well, your plasma membrane kind of can utilize those same properties. So if we want a more firmed up, if you will, plasma membrane, maybe in warmer temperatures or warmer conditions, we would have more saturated fatty acids as part of those phospholipids. If we were in a cooler environment and we don't want to be so rigid because we could actually fracture the plasma membrane, then we would have more of those unsaturated fatty acids. The one with the kinks gives it a little bit more flexibility. So the cell has the ability to respond to its environment and actually change that fluid nature to make it more rigid or less rigid depending on the environmental conditions. Okay, so the phospholipid bilayer is the major component of a plasma membrane. It's called the phospholipid bilayer, plasma membrane or cell membrane. So that's the major component, the ingredient, if you will. The next important or most abundant structure within the plasma membrane are what we call membrane proteins. So we got to think back to chapter 3, right? So our proteins were those big long chains of amino acids in a very particular sequence. Then they folded into either the helix, and then they folded again into that tertiary structure, and sometimes a couple of those came together to be the quaternary structure. So remember, that was just a review of proteins. So a lot of times when you're looking at pictures in the book, proteins are kind of illustrated as these big blobby things. So I like to call proteins big blobs. So that's kind of my connection to them. But they are not blobous in nature. They're very specific because of that three-dimensional shape kind of dictates their function. So a lot of these proteins are embedded in that phospholipid bilayer. So what that means is some of those amino acids in the middle part of that protein have to be hydrophobic, right? Because if they were hydrophilic, they would be repelled and not allowed to stay in the middle part of that bilayer. So for example, if we're going to take a look at this protein right here, we could assume that all of the amino acids, kind of right where I'm drawing this box, the amino acids that make up that part of the protein might have some hydrophobic qualities to them. Remember, those R groups had different characteristics, but then this portion here and this portion here, those are probably going to have some amino acids that have hydrophilic properties because now they're facing the watery environments. So that organization in that particular sequence of the amino acids and the folding of the amino acids allow them to be in that proper place. Can't do that. Okay, I was trying to erase those red lines, but apparently I can't. All right. So some of these proteins have different jobs. So the one that I kind of drew all over, this is a transport protein. It's going to allow things to move through the plasma membrane. We'll get to some of that a little bit later. Sometimes they can be a binding site, like a receptor, to communicate or receive a signal coming from another cell. Sometimes they can be anchoring proteins where they are going to be bound to parts of the cytoskeleton. So remember, actin, microfilaments, intermediate filaments, and microtubules. You see kind of the greens and purples there. Some of those are anchored to these proteins that are embedded in the plasma membrane. And then also some of these proteins might have carbohydrates stuck to them. So these little hexagons, right? So if you remember, glucose was primarily in the hexagon shape. So when you have a big branching chain of those hooked to a protein, it is called a glycoprotein. Glyco is a prefix that references sugar. Like glycogen was our storage of glucose molecules. And then the protein is what it's attached to, so glycoprotein. Okay, so those are the second most abundant characteristic or structure found in plasma membrane. So we have the phospholipids bilayer, and then we have all of these proteins embedded in them. Okay. So here's just a list showing some of those, kind of another close-up view of those proteins. So receptor proteins I already mentioned. Recognition proteins are like communication, let me give you an example of a recognition protein. So we have proteins on our surface that kind of have our name tag. So they're called self proteins. And so when our immune system cells are cruising around and they recognize our self proteins, they ignore us because they know that we are part of what we're supposed to do. If a bacteria or some other tissue or some other cell comes in, it's going to have a different protein on its surface. And our immune cells recognize that as foreign, and so it'll try to destroy it. And so that's where kind of the foundation for transplant rejections take place, or blood transfusion reactions where you have a problem, or it's how your immune system works to identify bacteria and viruses that have come into your bodies, because they have different recognition proteins than what we present on our cell surfaces. Some of the proteins can be enzymes. Again, we'll see enzymes more in depth in a future chapter, but just know enzymes are proteins that catalyze chemical reactions. They allow chemical reactions to take place. The attachment proteins connected to the cytoskeleton, and again, those transport proteins that allow things to move back and forth through the cell, which is going to be a bulk of what we're going to be talking about later in this lecture. Okay, so another key idea to membranes is that they're selectively permeable, also known as semi-permeable. You might see that term every once in a while or in a different resource. So selectively permeable means certain things can get through. Permeable means stuff can get through. Selectively permeable means only certain things can get through. It's not like all the doors are wide open and any old thing can come in and out. It's very selective. And the first part of that selectivity that we'll be seeing is the phospholipid bilayer itself. The second level of permeability regulation, that selective property, is going to be those transport proteins. Okay, so only specific substances are allowed across the phospholipid bilayer. So when we talk about things that can actually travel through that phospholipid bilayer, these are going to be small, non-polar, we could say hydrophobic as well, that they can just pass right through the plasma membrane. They don't need a transport channel. But anything else, anything that's large in size or polar, which means it has that charge on it, whether it's a straight ion, like a hydrogen ion or a potassium ion or chlorine ion, even though they're really, really small, they have that charge to them. And we know hydrophobic things repel anything that has a charge because they're nonpolar. So all of those tails, the hydrophobic tails in the middle, if you have a charged particle wanting to come in, it can't. It's a barrier. And so they cannot pass that hydrophobic region. Or if they're large in size, they're not going to be able to squeeze through. So if you want to like to say large and in charge, so large or with a charge. So if you are a large substance or polar, you have that charge to it, you can't get in or get out. And so you need to use one of those transport proteins, the ones that are embedded in the membrane that have kind of that tube or tunnel in the middle. Okay, so before we can start taking a look at the transport mechanisms, we have to do a little bit of groundwork here to set the stage for understanding things that are going to be moving through our plasma membranes. So first of all, we have to understand that all of the things that we're going to be talking about are taking place in a liquid environment. So the cytoplasm is like 90-something% water, so it's a watery environment. The fluid that you are in, whether you are an amoeba floating out in a pond or a river, or whether you are a cell within a body, you are surrounded by fluid. And so we have to understand when things are dissolved in fluids, they have some special terms for those. So here's some of the definitions of words that will help with our comprehension of these transport mechanisms we're going to get to. So the first word is called a solute. So a solute is a substance, anything that can be dissolved. Think of like salt or sugar or gases or ions or, anything. When you make salt water or sugar water, the salt or the sugar is a solute. The solvent is the thing that's doing the dissolving. So in our cases, that's mostly going to be water. So salt water, sugar water, salt and sugar are the solute. Water is the solvent, and water is a great solvent. We saw that back in chapter 2, I believe, because it is polar. It has that polar nature to it, and so it can dissolve things that are also polar. And then when you mix those two things together, solute plus solvent, it is called a solution. So we have basically our cytoplasm. It's not pure water. It is a solution. It is mostly water with a whole bunch of other things dissolved in it. Our environment is mostly water with other things dissolved in it. So we have a solution of stuff, like a mix of stuff inside of the cell and a mix of stuff outside of the cell. And that's important to understand because it sets the stage for the transport mechanisms we're going to talk about. So I found a kind of a very basic picture to illustrate this idea. So in this case, the blue dye molecules are the solute. The water is the solvent, and you put that dry blue powder in the solute, in the solvent, and you get a solution. We don't see the individual dye molecules anymore. It is mixed in, the water has dissolved that solute. Okay, so those are the solutions we're going to be dealing with: intracellular, inside of the cells, and extracellular, outside of the cells. Okay, so those are our solutions. So now we can take a look at concentration and concentration gradients. When we use the word concentration, what we're doing is we're trying to count or give some kind of number or value to how much of that solute is in a certain volume of solvent. How concentrated is your solution? Like when I make my hummingbird food, I put it on the porch, it's usually a 1-to-4 ratio. I have 1 cup of sugar for 4 cups of water. I usually don't make that much. I usually do a 1/4 cup of sugar for 1 cup of water, but that would be the concentration. It's a 25% solution. It's 1 part to 4 parts. So that's the concentration idea. Now, when we take a look at a gradient, the gradient is a difference in something. So we're going to be talking about concentration gradients, but you can have gradients in temperature, pressure, charge, And what we're going to be taking a look at is concentration. You might have seen things like when you watch the news and the weather, if you see like a high front or a low front, a high pressure system or a low pressure system, those are pressure gradients in the atmosphere, right? So that's the same type of idea. Things like to go from a high concentration or from a high gradient to a low gradient, where there's a whole bunch of something to where there's a lot less of it, whether it's pressure or temperature or concentration. Okay, so I found some pictorial representations of this. So on the bottom, I'm going to have you look at the bottom picture first. All of the dots are kind of evenly spaced. There's no real big like clumping of any of those blue dots in that bottom picture. This is what we call equilibrium. So when everything is kind of evenly distributed, there's no higher grouping of those anything in one particular place. That's equilibrium. Now, if you take a look at the top picture, you have over here on this side, you have a higher concentration. So all the dots are kind of all squished together over here very, very tightly. So that's a higher number of solutes per volume. Okay, so if I were to make a volume of just a square or a rectangle this big, right? So that's my concentration. And then if I come over here and try to make a rectangle about the same size, we can see within that same volume, there's a lot less solute within that volume. So what we're going to do, all right, so what we're going to be doing is comparing those two boxes. So we have the box on the right that has a higher concentration and a box on the left that has a lower concentration. So that's our concentration gradient, different concentrations of this particular solute, if you will, within this solution, the white space. So when we take a look at things wanting to move, we'll be introducing a term called diffusion here in a little bit, but this is just a universal principle. Things will go from an area of high concentration to an area of low concentration to try to reach equilibrium. This top picture, this whole picture right here, this is not stable. The molecules in the high end of this picture, they're bumping into each other. They don't really like to be so crowded. And if there's space for them to spread out, they will. They're going to kind of bump into each other and keep bumping into each other until they're all evenly spaced and they're all bumping into each other. And then you've reached equilibrium. So that's pretty much the goal for almost all of these transport mechanisms that we're going to be taking a look at. Okay. So here, this is a picture from your book. It's kind of the same idea, but specific here in a beaker. So this is this idea of concentration gradient and substances moving along or down their concentration gradient. And they do this, the question is, why do they move from one place to another? Is because if they want to go from that high, really crowded place to that more distributed equilibrium. So here in the picture down at the bottom, you have maybe a drop of food coloring, or a sugar crystal that's a sugar cube that has some dye in it, or just a plain sugar cube. It doesn't have to be colored. But you have the area of really high concentration there, and that's step one. And then just by random molecular movement, atoms are just bouncing around. That's what they do. And as they're bouncing around, they're going to move those dye molecules all over the place until eventually they're all equally distributed, and we are going to be at equilibrium. Now, we use this term dynamic equilibrium because once everything is spread out, they don't stop moving. They will continue to move, but now they're equally spread out. They're not going to ever go back to be in that highly concentrated drop of food coloring anymore. So they're still going to be moving around, but they're going to be maintaining that equilibrium. So dynamic equilibrium is constantly changing, constantly moving around, but still maintaining that equilibrium. Okay, so here I have a picture and a little GIF animation. You can kind of watch it. It's just going to go through multiple times. So this is introduced into the type of transport mechanisms. So there's two major categories that we're going to talk about. The first one is called passive transport. Passive transport is when a cell does not require any energy to move things across that selectively permeable membrane. The other type that we're going to be seeing a little bit later is called active transport. Active transport is where the cell actually uses energy to move things across that selectively permeable membrane. So there's three types of passive transport we're going to take a look at: is diffusion, facilitated diffusion, or simple diffusion, I should say, simple diffusion, facilitated diffusion, and osmosis. This slide is just talking about diffusion in general, and then we'll kind of differentiate between simple and facilitated here in a second. So the textbook definition, if you will, is when a solute moves from an area of high concentration to an area of low concentration to reach that equilibrium, that's diffusion. So that is our understanding. Hopefully you'll get a comfortable understanding of that term. So this idea of diffusion happens all over the place. It happens inside of cells, outside of cells, across plasma membranes, in the open ocean, in the atmosphere. Diffusion is happening everywhere. So things will diffuse. They will go from an area of high concentration to low concentration, trying to reach an equilibrium. You may experience this in your own day-to-day lives by, if you wear perfume or cologne or something like that, you spritz that on, and maybe you're standing in the bathroom and it's going to be really concentrated there in the bathroom as you're getting ready for your day. And then as you walk into a different room, it might be a little bit less strong. But then after a while, you go back into the bathroom or you're moving around in your day, and all of the scent is not going to be quite as strong in your original place of where you spritz that perfume, is because those scent molecules have evenly distributed throughout all of the space, and you're not going to have that strong scent anymore. That's diffusion. So it happens across a plasma membrane without any energy. This is our example of passive transport. And a lot of times we use the phrase, the substances or the solutes or the salt or the sugar or whatever it is you're taking a look at moves down its concentration gradient or with the concentration gradient. Think of it like a slide. You start at the top and you just slide down. You don't need any energy to do that. And so that's why you need to be thinking about diffusion as a passive, spontaneous, from high to low concentration. So we have in the picture, just kind of a static picture here, we start off with a high concentration on this side and a little concentration on that side, and you let some time go by, and if the membrane is permeable to those little red dots, they will eventually move and spread out equally both sides. And since this is a dynamic equilibrium, those red dots will be moving back and forth, but they will be moving at an equal rate. Okay? You will start off with more moving from the higher area to the lower area, but after a while they become equal, and that's our dynamic equilibrium. And then here in the little GIF, the animation, the left side starts off with a higher concentration, and by the time it ends, all the purple dots have evenly distributed to both sides of the membrane, and they'll keep moving back and forth. Again, dynamic equilibrium. Okay, so that's in general diffusion. Now we can differentiate between facilitated diffusion and simple diffusion basically by what it is that's diffusing. What is the solute? Now remember, these phospholipid bilayer, they're selective in just the nature of their chemistry, is that hydrophobic middle, right? All the little tails, they repel. They will not allow anything to pass that's large and has a charge. So things that can diffuse directly through the plasma membrane, we don't need a channel, a transport protein. This is what we call simple diffusion. It includes things like lipid-soluble substances, like vitamins, like A and E. Oxygen and carbon dioxide are respiratory gases. These are lipid-soluble, and they're very small. So these things have the ability to go directly through that plasma membrane following their concentration gradient. So they'll go from high to low. Now in this picture, it looks like they're at equilibrium. There's about the same number of oxygen molecules on the outside and the inside, so they have diffused equally. But that's what happens when we breathe. When we take in a lung full of air, we breathe in, we have a higher concentration of oxygen in our lungs and a lower concentration in the blood that just came back from the body. And so oxygen diffuses directly across our alveoli, the little sacs in our lungs, across your capillary and directly into your blood, into the liquid part of your blood and onto your red blood cells. So that's from a high to low area. And we're going to fill that blood up full of oxygen, and then it goes out to our body, and then we're out of our tissues. So now our blood has high concentrations of oxygen, and our cells don't, because they're constantly using it. So then there's another path of diffusion for high concentration of oxygen in the blood will diffuse directly across all of the membranes to get into the cell, so the cell can use the oxygen to make ATP in the mitochondria. All right, so this is what we call simple diffusion, small, lipid soluble or hydrophobic or nonpolar, whatever term you want to use. That is the characteristics that will allow things to go directly through that plasma membrane. Now, if you are not one of those things, so if you are large and charged or small and charged, if you have a charge or if you can't fit through that phospholipid bilayer, then you have to use one of those transport proteins. Those membrane proteins are embedded in the phospholipid bilayer, and a lot of times they're very specific. So we have the one on the left for glucose. Again, glucose is almost always going to be shown as that hexagon shape because it's kind of a nice, easy shape to show that sugar. So they cannot pass directly through the phospholipid bilayer. They're going to be repelled by that hydrophobic middle part. So they have these different kinds of channels. There's a couple different types that can be used. The first one is called a carrier protein. So I like to think of a carrier protein as almost like a Pac-Man, right? My face out of the way. So it opens on one side, there we go, to the outside of the cell. And say the glucose comes in and sits, and then it goes Pac-Man, and then the glucose can go inside, and then it's going to change. Glucose will come in, Pac-Man. So it kind of does this. It's passive. It's only going to do that if the glucose is going down its concentration gradient. So in this picture, we have a lot more glucose outside of the cell than inside of the cell, and it will continue to do its little Pac-Man until it's reached equilibrium. The other type of facilitated diffusion, this membrane protein, is called a channel protein. So channels are just open. They're just like a gate, not a gate, like a tunnel. And so they're always open. They can be specific. So this one's specific to chlorine. If there's one for sodium, it wouldn't allow the chlorine through. So they're open, but they're specific for their solute that's going to move through, but they don't change shape. So carrier proteins change shape. Channel proteins stay open like a channel, like a tunnel, through that phospholipid bilayer, allowing those large or charged particles to diffuse, again, down their concentration gradient. So we can see in that picture on the right, there's a higher concentration of chlorine outside of the cell compared to inside. So it will continue to move through until it reaches equilibrium. Okay, so that was our first type of passive transport, or the first two types, so simple diffusion and facilitated diffusion. Those processes deal with solutes, salts, sugars, vitamins, ions, electrolytes, all of those different types of things. This last type of passive transport is called osmosis, and it is specifically dealing with water. It's the diffusion of water. That's its definition. So osmosis is the diffusion of water across a selectively permeable membrane, again, to try to reach that equilibrium. That's the law of the universe. Everything's trying to reach that equilibrium. Again, it's passive. It occurs spontaneously and relatively quickly at that. Now, when students are learning the movement of water in osmosis, there are a couple different ways that they can take a look at it. So I'm going to present it in two ways. You can pick which one sits better with your understanding. Some people like to stay high to low with the diffusion because of the diffusion, but other students like to take a look at it as how water moves in regard to the solutes. So these are the two ways that you can kind of think of osmosis, or when you're trying to figure out the directionality, which direction is the water going to go? Is it going to go inside of the cell? Is it going to go outside of the cell? This is how you figure that out. So one, because it's diffusion, things go from high to low. So you can take a look at where do you have more water and where do you have less water, right? It's a concentration gradient. Water will move from the area of high water to an area of low water, right? That's a good definition. The other way to think about it is water tends to go towards the area of higher solute concentration. There's a saying out there in the world of water follows salt, or water follows solute, because it doesn't have to be salt. It could be a sugar solute as well. So water follows that higher solute concentration. The reason why these things are the same is because if we take a look at it, what if you have one side of the cell is pure water, 100% water. So it's the area of high water concentration and very little, no solute. And then on the other side of this membrane or inside of the cell or a different area, you're going to have maybe 50% water and 50% solute. Okay, so you have 100% water over here and 50% water over here, water's going to want to go from the high to the low water concentration, right? You're going to have water go from this side to this side. Let me get my hands here, right? So if we have a whole lot of water over here, 100%, and 50% here, water will go towards that lower water concentration, okay? You can think of it also as the higher solute concentration. So if this is 100% water, 0 solutes. And if this is 50% water, what's the other 50%? Solutes. So water will also go towards the higher solute concentration. So you can see my hand is moving in the same direction from high water to low water, 100 to 50%, or towards a higher solute concentration from 0 solutes to 50% solutes. So your choice. You can, if it makes sense both ways, use both ways. If you like the high to low water, use that. If you like water towards the higher concentration of solutes, use that. Use them both. Just pick whichever one works best for your brain. I like to use the water follows salt just because that's a little bit easier to figure out, because then I don't have to figure out the water concentration. I just look at where you have more solutes, and that's where the water is going to go. All right, so here is a static picture and a little animation showing this idea of osmosis. So I'll talk about the static picture first. So this beaker here, on this side of the beaker, we have a lower concentration of substance, right? And then on this side of the beaker, we have a higher concentration of substance. And you can just see that by the purple dots in the pink fluid or whatever. So if we are going to take a look at it, either of those ways to take a look at it, we'll start with that first definition, that first way to try to figure out the directionality. On the left-hand side, you have high water and low, I'll just do S for solute since I'm not a very good PowerPoint writer. So on the left-hand side of the beaker, you have a lot of water and a little bit of solute. On the right-hand side of the beaker, if you take a look, if you have more solute, you have less water. So you can just look at the pink stuff. You have a lot more pink space on the left and a lot less pink space on the right. So you have less water and you have more solute, right? So if we're going to follow our definitions of water or osmosis is the diffusion of water, we can go and utilize both of those things, right? Both of those mechanisms of thinking. So if we have a high water over here, that arrow is horrible. There we go, a little bit better. So water will go from an area of high to low water, okay? That fits. Or we can have water go towards the higher solute concentration. That fits. They're both going in the same direction. It's not going to give you different answers. So if we let time go by, what will actually happen if you were to be able to set up this experiment is the water on the right-hand side of this beaker, this separated beaker by the semi-permeable membrane, the purple solutes can't move through, just the water. So it's impermeable to the purple solutes, permeable to the water. Water will actually push against the gravity and actually make the water on that right-hand side go a little bit higher to try to reach an equilibrium. So now if we take a look at the concentrations between both sides, those purple circles are equally distributed around the pink water, okay? So this is our idea of osmosis. Water will go from high water to low water, or water will go towards the higher solute concentration. Now we can take a look at the GIF. I know it runs pretty quickly. We'll kind of watch it through a couple cycles. So when it starts, we have a whole bunch of water. That's the little blue circles. So we have a whole bunch of water on the left-hand side. And if we take a look at the right-hand side, we have the green circles are the sugar. So we have more solutes. So here's your kind of, they have it written out. So low sugar versus high sugar. It really should be no sugar because there's no sugar on that side. Right? And we have high water to low water. So we're going to get the same direction. Water likes to go from a high to low concentration, or water likes to go towards the higher solute concentration. And it's the same idea depicted in the picture at the top, but you're actually seeing the water level rise as those water molecules are moving to try to create an equilibrium on that right-hand side where you have a lot more of those solute molecules. All right? So those are kind of our ideas of osmosis. Now, with that being said, if you are still struggling with those ideas, maybe pause. You could pause the video right now and go back and reread the textbook, or try and make sure you're getting an idea and being comfortable with that idea of the directionality. Where's water going to want to go? Because when I introduce these next terms of tonicity, this is where things can kind of get turned around a little bit. So do what you need to do to make sure you're comfortable with those ideas of osmosis and the directionality of where the water is going to go before you come back and take a look at this tonicity. All right, if you're ready to go, let's go. So we have some special terms when we're talking about tonicity. It refers to kind of relative concentration gradients. You're comparing one environment to the next. So it refers to the concentration of solute relative to water concentration between two different environments, okay? So a lot of times we see it in relationship to the cytoplasm, the inside of the cell versus the outside of the cell. You'll see those scenarios mentioned a lot in these tonicity questions. So let's imagine your cell, right? And you're floating in your environment. So you have your plasma membrane, you have all the cytoplasm inside of you, and you're floating, maybe you're an amoeba or a paramecium floating out in the pond or something. If the concentration of things inside of your cell is exactly the same concentration as the fluid that you're floating in, we have a term for this called isotonic. Iso means the same. And tonic has to do with this tonicity, this concentration gradient comparison. So if you're same on the inside compared to the outside, you're called isotonic. You might have osmosis happening, but it'll be at equal rates. So water will move in, water move out, but there is a dynamic equilibrium going on. If you're the amoeba, you're not going to shrink, you're not going to swell, you're going to stay the same. You're going to stay equal. Now, if your solution that you're floating around in has more concentration of solutes compared to the cytoplasm, so now you have a whole bunch more stuff out here than you do in here, this environment is what we call hypertonic. Hyper meaning high. Think of like hyperactive. So hyper meaning hyperthermia, your temperature is too high. So hypertonic is more solute concentrated compared to another environment. Oh, my face is kind of taken over. So if the solution has less concentration than the cytoplasm, it is called hypotonic. So hypo is a prefix that means less than. So think of hypothermia or hypothyroidism, some women and men, but more common in women. Some people have hypothyroidism is because their bodies are producing not enough thyroid hormone. All right, so those are our three tonicity terms. Iso, the same between your two environments. Hyper, one has more solute than compared to something else, and hypo is less solute compared to something else. So let's take a look at some examples to see why this is important. Okay, so I have, your book has a picture of just the red blood cells, and then slightly, it has some of the plant cells too, but I wanted to show them kind of all in the line with the difference between these solutions. So this picture shows the red blood cell, which would be like an animal cell on the top, the plant cell on the bottom, but they both kind of respond the same way. So let's take a look at that first box. This is the isotonic solution. So again, remember isotonic means same concentration. So in the animal cell, water will move in at the same rate water will move out. And in the plant cell, it's the same idea. You're equal concentration. So osmosis is going to happen, but there's no net gain of water and there's no net loss of water. Okay. So in the middle picture, you'll notice they kind of did the shading a little bit. They started with a medium shade of blue, and then in a hypotonic solution, which is the blue box, all of this is the environment out here, right? That's the environment. So a hypotonic solution means the cells have more solute in them compared to the outside. The outside has less solute compared to the inside. So it is hypotonic solution. Now in that case, water likes to go where? Right? You had those two choices to kind of verbalize it. Water can go from high water to low water, or water can go towards the higher solute concentration. Now, if you're in a hypotonic solution, you have less solute, which means more water, and the cell has more solute. That's just the nature of it being in a hypotonic solution. So water is going to move into the cell, and it will continue to move into the cell until what? Equilibrium. So water will continue to move inside of the cell until a couple things might happen, depending on which type of cell you are. If you are an animal cell and you have a really hypotonic solution, think like distilled water, water will continue to move in, and we don't have a protective cell wall. We just have that phospholipid bilayer. It can only resist so much influx of water. And so what happens is the cell bursts, or what we call lyse. So cell lysis happens in animal cells. In plant cells, we learned about that large central vacuole, right? Stores a lot of the water. So the water will move in and it's stored in that vacuole, and the vacuole kind of pushes up against the cell wall. And this gives our plants kind of their turgid, upright stance, if you will. So imagine the last time you didn't water your plants, what happened? They kind of get wilty. They get wilty because you don't have all of that extra water in the central vacuole pushing up against the cell wall. And you could have a wilty plant here in an isotonic solution, right? And you will definitely have a wilty plant over here in the hypertonic solution. So your book has the pictures of those plants on page 82. So you can kind of take a look at the comparison of how that central vacuole looks in an actual, in the textbook. All right, so in the last box here, so here's the environment out here. It's a darker blue. So this is our hypertonic environment. So it's in comparison to the inside of the cell. Remember, hyper has more. So more solute inside of the cell, more solute in the environment compared to the inside of the cell. So again, water is going to go towards the higher solute, or from high water inside of the cell to less water outside of the cell. In either kind of phrasing, water is going to leave the cell. And in an animal cell, there's no cell wall, so the shape is actually going to shrivel up. So this, you can kind of see the arrow of the water leaving the cell. And in a plant, it will leave the central vacuole. It still has the cell wall, so it has a little bit of structure to it, but it's going to be really shriveled and wilty. So that's going to be like maybe if you accidentally put some salt water on your plants, then you make all the soil and the water around the roots hypertonic, and all the water leaves the plant trying to go create an equilibrium with the salty soil, and that's no good for keeping things alive. All right, so when we talk about these tonicities, isotonic, hypotonic, hypertonic, this is the idea of where water goes. It's like I said, it's spontaneous, it occurs very rapidly. So water will move in response to these different environmental conditions. So we want to be isotonic. We don't want any net gain or loss of water in our cells. And you may know, hopefully you know, just by hearing things live in your life, you don't ever drink seawater, right? Ocean water is about 3.5 times more concentrated than our cells. We are about 1% salt, and it's about 3.4% salt. So if we were to put a salt solution drinking salt water, we're basically bathing our cells in this solution here, in the hypertonic solution. So what's going to happen? All of the water inside of our cells is basically going to get sucked out. Our cells are going to shrivel and they could die. Leads to severe dehydration, so don't drink salt water. But could we do damage if we were to drink too much fresh water or distilled water? What do you think? Certainly. What if we were to drink a whole bunch of distilled water? We would then be flooding all of our cells with a really hypotonic solution, and our cells would be the hypertonic. Our cells would have the higher solute concentration, and water can flood in, and we don't have any protective mechanism against that, and our cells would burst. It's actually a condition. It's called water or overhydration. Drinking too much of the pure water, your kidneys can't keep up with that, and you can actually cause some serious organ damage, and it's fatal if you drink too much of that distilled water. So don't do it. No salt water, don't drink distilled water. It's not good for you. And this is why. Now you know why. Okay, so those mechanisms of transport are what we call passive. The cell does not expend any energy for that. It is a passive process. Things just like to go down their concentration gradient. It happens freely. But there are sometimes a cell needs something that it already has a lot of. It needs to keep bringing things in, like glucose or amino acids, things that it needs to build more of itself or to burn for energy, right? We use glucose for energy. So in that case, the cell will actually expend energy to pump things against their concentration gradient. Okay? So this is called active transport. And so if we want to kind of put it in a definition similar to diffusion, we can say active transport is when a substance moves from an area of low concentration to an area of high concentration. Now we don't have that equilibrium word at the end of this definition because we're not trying to reach equilibrium. We are going against equilibrium. We are moving things, think of that blue dotted page that I had. We are trying to pump all those dots back over to the pile, and that takes energy. A good example that I like to say when I'm talking about transport is the passive and active. Think about it's summer, right? Maybe you're gonna go rafting up at Shady Cove if you live here in the Rogue Valley. And so you hop on your raft way up by the dam, by the lake, and then you passively float down a couple hours, 3 hours down to Shady Cove. You don't have to have a motor, you don't have to paddle. That is a gravity gradient that you're taking advantage of. But what if you hop on Shady Cove and you want to get up to the hatchery or the dam by the lake? You can't just float upstream. You have to put in energy. You either have to paddle or kick or have a motor to actually go against that gravity gradient of the water. So think of active transport as trying to swim upstream or paddle upstream. It takes energy to do that. It does not occur spontaneously, right? So you have to actually have some energy input to do this. But it's also very common. This is something that's not just a rare event. It's happening in all cells, bacteria, animal cells, plants, fungi, all cells are trying to get things that they need to maintain their homeostasis. All right, so here's an example that your book presents of active transport. This is a common mechanism in cells called calcium pumping. So usually we use the word pump when it's active transport as opposed to a diffusion. We don't really use the word pump with diffusions because think of a pump, you have to use energy to pump stuff somewhere. So here, if we take a look at the first panel right here, we can take a look, identify your concentration gradient, right? The blue circles, those are the chlorine atoms or ions, Cl2 plus, or keep saying chlorine, calcium, I should say. There is the higher concentration in the pink area, that's your interstitial fluids. That's outside of the cell. The blue area is inside of the cell. So if we have more calcium outside of the cell and less inside, that's our concentration gradient. And if we're going to be doing active transport, right, active is going against that gradient. So we are wanting to move more calcium outside of the cell. So here we use a cellular energy molecule called ATP. We'll be learning more about that a few chapters later. So ATP is used. So it's kind of like that Pac-Man, right? But instead of being a carrier protein moving down the concentration gradient, we're going to be binding ATP and the calcium on kind of the inner surface here. And then with the power of ATP, it's going to force that pump open, and then the calcium is pushed out. And then we are going to release that phosphate group, and it changes the shape back in to take in another calcium. And it's kind of like that Pac-Man, but it requires energy because we're kicking out the calcium into an area where there's already a whole bunch of calcium, okay? So this is our example of active transport, okay? The last few types of active transport are more large scale kind of bulk transport mechanisms. They're usually going to be doing, cells are gonna be doing these things for larger, like they're so big they can't fit through any kind of transport protein. So the first type we're going to take a look at is called endocytosis. There's endocytosis and exocytosis. So endo meaning kind of internal, cytosis like in cyte, CYT means cell. So we're bringing something into the cell. So there's three types of endocytosis, three ways that stuff is being brought into the cell. We have pinocytosis, receptor-mediated endocytosis, and phagocytosis. Okay, so pinocytosis basically means cell drinking. So this is where the plasma membrane out on the surface kind of folds in, makes these little dents, and just kind of starts to continue to fold around the interstitial fluid, kind of bringing in a vesicle's worth of the environmental fluid. And so you can kind of see that in the drawing here, and you can see it in the electron micrograph here, the little folds pinching off, and then you can see the vesicle that's been brought into the cell. So pinocytosis is bringing in fluid stuff from the outside of the cell to the inside. The next one is called receptor-mediated endocytosis. And again, it's bringing in things that are too large to transport through the plasma membrane, but this is a lot more specific. The reason why is this receptor-mediated. It's the receptors that have to bind to a particular substance. So just not any old thing can trigger this process of endocytosis. So in the picture, the thing that we want to bring in is this green ball here. And so the green ball has to bind to a very specific site on the surface of the cell, those little blue Y-shaped molecules. And so if it was a different molecule, maybe a red square, it wouldn't fit in those little receptors. And so it wouldn't be brought in. So there's a lot of specificity with this receptor-mediated endocytosis. And so once the thing that the cell wants in is recognized by that receptor, it triggers that folding in to this vesicle, and now you've just taken in that thing that you want. Here's a cool thing about receptor-mediated endocytosis. Cool for viruses, not for us. So a lot of times viruses or sometimes bacteria fool our cells because they have adapted and acquired molecules on their surfaces that mimic those signaling molecules. Those, what are they calling? Just molecules. And they can bind to those receptors, and the cell's like, oh, I want that thing. I have these receptors out here to bring it in. And so here comes a virus docking instead of the substance, and then the cell brings it in, and voila, our cell has just been infected by a virus. So they're sneaky little things that have been able to take advantage of how our cells would normally take in substances. HIV, the virus that causes AIDS, does something very similar to this to get inside of our white blood cells. Okay, the last type of that endocytosis is called phagocytosis, which basically breaks down to cell eating. Phago means to eat. So usually this is seen in protists like an amoeba or our white blood cells. Some of our white blood cells do phagocytosis, and that they can take in even other organisms, so large substances like food bits. But sometimes for an amoeba, the food is another organism, like a smaller paramecium or a bacteria. So here you can see the food particle, which could be a bacteria or a paramecium or just a chunk of food, gets enveloped in the plasma membrane, pinches off, and now you have a food vacuole, which will then go infuse with a lysosome, right, chapter 4 stuff, to digest that whatever was brought in, like the bacteria or the parameciums or something. So here you can see a picture of an amoeba. That's the green blob here. And the pink little critter, that's going to be its food. So it's going to send those, what are called pseudopods, these little extensions of the cell cytoplasm around it, and consume it. And in the picture over here, this is a bacterium. The orange thing is the bacterium. And then this big brown thing here, that's the white blood cell. So it's extending its pseudopods out around to engulf that as well. I have a video, links on this week's page of a really cool video. It's less than a minute long, but it actually shows real-time footage of an amoeba eating 2 parameciums. It's very cool. You'll see the the phagocytosis making the food vacuole, and you actually see what happens when it fuses with the lysosome. It's pretty cool. Just know the paramecium have no pain receptors. They're sensory, they're responding to their environment, so don't feel bad for them. It's part of the circle of life, right? So enjoy that video. Okay, and so our last type of active transport we're going to talk about is called exocytosis. So it's almost a reversal of endocytosis. It is when a cell needs to get rid of large substances relatively quickly. This could be a cell getting rid of waste products. Maybe it took in the bacterium, it digested it, it has some bits it can't use, and so it just wants to release that back into the environment. It could be a cell secreting any of its cellular products like mucus or tears or saliva or oil or, whatever. We have a lot of secretory cells in our body, enzymes, hormones, those types of things. And so basically, if you remember back in chapter 4, substance might be made on the rough endoplasmic reticulum, packaged in the Golgi, put into a vesicle, sent to the surface, and you'll see in this picture, the vesicle fuses with the outer plasma membrane and then just releases the contents into the outside world, all right? So pretty straightforward, exo meaning outside of the cell. So all of these, the calcium pumping, the endocytosis, the exocytosis, these are all active transport mechanisms Again, remember, meaning that they require cell energy to do that. We don't see the direct activity of that activity of the ATP, but it is involved in the endocytotic and exocytotic processes. All right, so this last slide here is just a summary. You have this table very early on in the chapter, but I wanted to put it here at the end after we've covered all of these things. So this is a good summary table for you to take a look at and review the different types of passive transport: simple diffusion, facilitated diffusion, osmosis, and then our different types of active transport, the pumping, endocytosis, and exocytosis. So use this table for review. Go back and make sure you have a good understanding of diffusion, osmosis, active transport mechanisms. So we are done, and I will see you next time.